Shruti, as Jenny mentioned, I'm the coordinator of the ClinGen Somatic Cancer Cdwg. Deborah, Ritter, who coordinates the Cancer Variant Interpretation Committee, and I will start off with giving you an overview of the ClinGen Somatic Cancer Cdwg, our somatic cancer variant curation expert panel process, and then the Cancer Variant Interpretation Committee that was formed to help the somatic cancer Cdwg as well as our VSEPs. So the main goal of the ClinGen Somatic Cancer Cdwg is to standardize the curation and interpretation of somatic variants in cancer by uniformly implementing practice guidelines. And we do this by collaborating with various experts from different organizations to develop processes that support the accurate determination of the clinical significance of somatic changes. And our ultimate goal is to enhance the usability, dissemination, and the implementation of our somatic variant curations in the ClinGen resource, as well as other knowledge bases like CIVIC, ClinVar, GA4GH's variant curation and expert variant interpretation in cancer consortium. So that's the VIC, their meta knowledge base, as well as for discussions in virtual molecular tumor boards. So briefly, our somatic cancer Cdwg has over 100 members from 30 different organizations, ranging from academic medical centers to LDT developers, from the government, government agencies like NCI and FDA, as well as other partner research consortiums and knowledge bases like GA4GH, VIC, and CIVIC, and then professional organizations like AMIA, AMP, ASCO, CAP, and ACMG. All of our curation activities within the somatic cancer Cdwg take place within three main disease areas: pediatric cancers, hematologic cancers, and genitourinary cancers. And one of the main goals of these disease task forces is to assess candidates for future somatic cancer variant curation expert panels. So we currently have two SC VSEPs that have received step 1 approval from ClinGen. The NTRK Fusions SC VSEP was our very first somatic expert panel, and it came out of the efforts of the Pediatric Cancer Task Force. And the FGFR genes VSEP recently received step 1 approval, and that's from the Genitourinary Cancer Task Force. The Hematological Cancer Task Force will soon also be submitting a VSEP application for FLT3. So our Cdwg is co-chaired by Dr. Shashikant Kulkarni and Subha Madhavan. And these are our disease task force and expert panel co-leads. In the third row here, you'll see our informatics leadership. Sorry, I'm just gonna move this out of the way for now. So in 2016, the Somatic Cancer Cdwg published the minimum variant level data framework. And this framework, the main goal of this framework was to provide a framework for curation and reporting of somatic variants in cancer. So briefly, the data framework has 3 main categories: the allele descriptive, the allele interpretive, and the somatic interpretive. The allele descriptive category has information about the gene, the chromosome, DNA position, the reference transcript, and the reference protein ID. The allele interpretive section has information on the DNA substitution and position, the protein substitution position, the variant type, as well as the variant consequence and any reference PubMed IDs. And the somatic interpretive section is the main section that has most of the information to help with classifying the clinical significance of these variants, starting with cancer type. And we provided ontology recommendations for the cancer type. the biomarker class, so whether the variant has a diagnostic, prognostic, or predictive clinical significance. For predictive variants, we also provide therapeutic context. So what drugs or drug classes are associated with the variant. And then the effect of the variant, so that's the therapeutic context again. So whether the variant predicts resistance to a drug, response, whether it's not responsive, sensitive, or reduced sensitivity. When we published these guidelines in 2016, we published them with our levels of evidence based on consensus from members of the somatic cancer Cdwg. At that point, we were also aware that AMP-ASCO-CAP was developing their guidelines for the interpretation and reporting of cancer sequence variants. So in 2017, when AMP-ASCO-CAP published their guidelines, we incorporated them into our levels of evidence module. So I assume many of you are already familiar with the AMP-ASCO-CAP guidelines, but for those that are not, I'll just briefly go over each one of these tiers. So the tier 1 and 2 in the AMP-ASCO-CAP guidelines so tier 1 relates to variants of strong clinical significance, and tier 2 is variants of potential clinical significance. Within both of these tiers, we can interpret variants within their therapeutic, prognostic, and diagnostic effect. Within tier 1, level A evidence is for variants that predict response to drugs that have an that are FDA approved for a particular cancer type. And there's a companion diagnostic associated with them to detect the particular variant. And the information is also included in professional guidelines like the NCCN. Level B evidence comes from well-powered studies with consensus from experts in the field. And an example of this would be the Ikaros deletion that's associated with poor prognosis in B-cell acute lymphoblastic leukemia. So in terms of increased risk and relapse and decreased event-free survival. So that would be, we would classify such a variant as tier 1, level B prognostic. And then in tier 2, there's level C and level D evidence. Level C is for FDA-approved therapies in a different cancer type. So a cancer type that is not of your interest. So an example here is the drugs encorafenib and binimetinib that are used for treatment in melanoma patients that have the BRAF V600E and K mutation. And these drugs are FDA approved for melanoma, but then they're in clinical trials right now for pancreatic cancer patients with the BRAF V600E mutation. And then finally, level D evidence within tier 2. This evidence comes from preclinical studies and a few case reports. And then tier 3 evidence is includes variants of unknown significance, clinical significance, and then tier 4 is benign or likely benign variants. And these are variants that have been observed at significant allele frequencies in population databases. And there's also no existing published evidence of cancer association. So at this point, we're going to launch our first poll question. And we'd like to know, what guidelines do you all use to curate and interpret somatic variants? Is it the AMP-ASCO-CAP guidelines? For our audience that maybe in Europe, do you use the ESMO scale for clinical actionability of molecular targets, or do you use other guidelines that have been developed by your institutions or by partner institutions?
And can we also ask, if you do use other ones, could you put them in the chat so that we can know what it is that you're using?
Yes, that would be really helpful.
Another little bit of time
Mhm.
to log any more responses. Looks like everyone has answered, so I'm gonna share the results.
All right. Well, that's good to know that most folks use AMP ASCO CAP. For those that answered other, it would be great if you could let us know what guidelines you use in the chat box. All right, so moving on. A little bit about our disease task forces. So like I mentioned before, we have 3 disease task forces, pediatric cancers, hematologic, and genitourinary. The pediatric cancer task force was our very first task force that was formed in 2017. It's co-led by Drs. Anshumoy Roy and Gordon Araka, and we have now grown to 61 members in this task force. The main goal of the task force, looks like I have an automated timer set on this. So the main goal of the pediatric cancer task force is they shortlisted 44 highly mutated pediatric cancer genes across 23 tumor types. And their goal is to curate the variants in these genes, but also identify potential SC or future SC-VSEP candidates. The Hematologic Cancer Task Force is co-led by Drs. Rashmi Kanagal-Shamana and Sinji Shu. This task force is a new task force that was formed in 2020, has 48 members to date, and they're mainly focused on the curation of FLT3 variants. And other genes of interest for this task force are also the BCR-ABL kinase domain mutations in CML and BCL2 mutations associated with venetoclax resistance. The pediatric cancer and hematologic cancer task force members also plan on collaborating to form a VSEP in PH-like BALL. And then lastly, the genitourinary cancer task force has 12 members, is also newly formed in 2020. It's co-led by Dr. Shamini Selvaraja and Ian King. And their goal is to curate variants in the FGFR family of genes. And they've received step 1 approval for this. So like I mentioned before, one of the main goals of the disease task forces is to form somatic cancer variant curation expert panels. And for this, they follow the 4-step process that you see on the screen here. This process was modeled after the current ClinGen germline VSEP process that is FDA recognized. So briefly, the first step is defining the scope and the goals of work. So here, the somatic cancer community would basically prioritize or would identify the potential VSEPs, and then, and then we identify the VSEP membership and leadership, define the scope of work, and address potential conflicts of interest. The step 1 application is first submitted to the Somatic Cancer Cdwg for pre-approval, and then is finally submitted to the Clinical Domain Working Group Oversight Committee for their approval. Step 2 is where the VSEPs will develop variant classification rules. And we basically start off with existing guidelines. So AMP ASCO CAP, the MVLD. The somatic cancer group uses the Civic, which is an open access curation platform and cancer knowledge base. So we use Civic for curation, and so we review our VSEPs, review their SOP as well. And then we come up with guidelines to identify further areas of specification. If applicable, we also will try to develop computational approaches to help with the variant classification. Throughout step 2, our SC-VSEPs closely communicate with the Cancer Variant Interpretation Committee, or the CVI, and Deb will give us more details about the CVI and their goals and their current activities. So step 2 is then submitted to the CVI. And once we receive approval, we go into step 3, which is the pilot curation phase. And here we'd curate, oh, sorry, was there a question? All right. So step 3 is the pilot curation phase, where we'll curate a short list of somatic variants prioritized by the SCVSEP to validate the rules that were developed in step 2. These, again, the results from the pilot curation will also be closely communicated with the CVI and the somatic cancer Cdwg. And finally, the materials will be submitted to the oversight committee for approval. And then step 4 is defining plans for ongoing variant curation, review and reanalysis, as well as discrepancy resolution. And once step 4 is complete, that application would also go to the Cdwg Oversight Committee for final approval. We have also, so we are also interested in seeking FDA recognition for the somatic cancer expert panel process, similar to what's already been done on the germline side. We did submit a pre-submission inquiry to the FDA in April, and we're hoping to talk with the FDA reviewer sometime in the summer, and then decide on next steps. So with that, I'll turn it over to Deb to discuss the CVI.
Great. All right. Thank you. I will share screen. All right. Can you see the slides?
Yep.
Yes.
Great. Okay. Let me put it slideshow. What do you see now? Just the main slide, not the presentation mode?
We see the presentation. Oh, the main slide, yeah.
The main slide. Okay, great. All right, so I want to talk some about the ClinGen Somatic CVI. This is a recent development in the somatic working group. The chair is Shashikant Kulkarni, and I'm the coordinator. So this was approved by the ClinGen Steering Committee, and it will work closely with the SVI as well as the cancer VSEPs to support the ClinGen somatic variant curation expert panels. So, you know, I put here that this kind of bubble that we're thinking of the SVI, really we're trying to model a lot of our processes after the SVI and apply it to somatic variants and somatic variant curation expert panels. Let's see. Sorry. There we go. So we had our first introductory meetings in October of 2020 and our first official meeting in November. So it's quite recent. And we have recurrent meetings on the first Thursdays of every month. And this is our membership. We have about 26 members across many different institutions and also areas of expertise within cancer variant interpretation. So our aims are to explore some generalizable elements of the AMP-ASCO-CAP guidelines, similar to what the SVI does with the ACMG guidelines, to provide guidance where specific criteria will be needed, lead towards semi-quantitative approaches for variant interpretation, to tackle broad problems such as missense predictors for activating variants, work closely with these initial somatic cancer VSEPs in the same process. So some of the members of the somatic cancer VSEPs are on the CVI. And so, you know, there's a little bit of overlap there that we navigate, but I think it's very important to have that feedback for the CVI as well. And then to give input and to approve on cancer or gene-specific guidance for the VSEPs. So this is our timeline. We have started with a couple of projects that were in progress before, somewhat before the CVI developed. This oncogenicity SOP, which is you know, really creating some of the foundations for further specifications. Then fusion oncogenicity as well. The INTRACT fusion VSEP was in quite good progress. And so we've had them present to the CVI just in April. And with a little bit of feedback, I think actually they'll be moving into their pilot around June. Then the somatic hematologic VSEPs and genitourinary, I actually didn't put that in here, And then the somatic CVI here has established monthly meetings underway. And we do also intend to be a part of the FDA submission process, kind of in a similar way that some of the development from the SVI informed the FDA process for the germline side as well. So currently we are working on oncogenicity of fusions and of variants. So this would be general guidance here, and oncogenicity interpretation for variants. So this would apply to non-fusion, non-structural variant. And this is a manuscript that is in development and quite far along. And then working with AMP ASCO CAP, really to ensure continuity across any, general specifications we may make and any further additional rounds of guidelines coming out from these groups. So this was the initial standards and guidelines for the interpretation of reporting sequence variants in cancer. And we had some introduction to that from Shruti. But I think, a general point here is that similar to how the ACMG guidance came out, and there were areas that needed further specification, that's the same status for somatic variants currently. So in these guidelines that came out, there's still quite a lot of room for interpretation and specification as well. So just some examples here, for level A evidence, it says it has to be included in professional guidelines. It doesn't specify, which professional guidelines, or do some guidelines matter more for other cancers and other types than others would. Here, for instance, well-powered studies with consensus, there's been a lot of questions about what this means, about how to define this, what types of studies are needed, what does consensus include? Does that mean you need to have a minimum of 2 papers that have reported on this, and questions around that. For level C evidence here, you could ask, what is an acceptable range of tumor types, or define that? What are some of the elements in studies that are required? So if you do have these small published studies, and I would say some of this would be very similar to some of the functional guidance that has come out from the germline side. And again, lack of consensus here, trying to define that here as well. So these are just a few of the questions. There's a lot of questions available. For the unknown clinical significance, defining prevalence in population and defining benign allele frequency. Many cancers can be quite common. So I think lung cancer is about 6% in the general population. So just defining that when you're working with some of these cancers that are really not so rare in the general population. So trying to define these allele frequencies and defining which databases are required to use or acceptable for use. So again, just a selection of the questions that we will be working on. So for cancer variant interpretation progress, we've had a lot of progress, and there's a lot of interest in moving forward with this group. So we have already approved a manuscript concept sheet, which was Dmitry Sankin has really led this work for a while now. So it's quite near publication when we did approve the manuscript. But it's for the process of interpreting oncogenicity of somatic variants. So I think it's a very exciting development, and looking forward to having that come out. So I think we plan that it will create the foundation for additional specifications and additional work that will be done by the CVI. We have already provided 1 round of feedback for the first somatic VSEP, so in-track fusions. And in that, we had to develop the feedback process and our mechanism, internal to the CVI for providing that. And then our next steps coming up will be several projects and analysis efforts coming up. So, and many of these, I think, also came to light through the oncogenicity manuscript. So evaluating in silico predictors for missense activating variants, modifying some of the germline functional workflow into some decision workflows for somatic cancer functional data, modifying the PVS1 decision tree for somatic cancers, clinical trial interpretation guidance, so providing guidance on how to interpret some of this, as well as defining consensus across multiple groups, and then also looking at population databases and activating oncogenes and identifying appropriate control sets for somatic variants. So I think we have a lot of exciting projects coming up. And we do have a poll question. And while that poll question is up, I will leave a shameless plug for any interest in getting involved with some of these projects. So the poll question here would be, what areas would you like the CVI to further work on in terms of the areas that you think matter in the next steps that we take? I don't get to answer the poll, but obviously I think they're all important.
Like the responses are slowing down. We'll just give you a few more seconds to add responses.
Sure. Great.
All right, I'm going to share results.
Nice. It looks like functional and the guidance on consensus and that. Wonderful. That's good to know. Thank you. All right. Shruti, I will hand it back to you.
All right, great. Thanks, Deb.
You have to just stop sharing. There we go.
There we go. All right. Actually, before I move on to my slides, Jenny, can we launch the third poll question as well, please?
Yep, sure.
Thanks. Okay, great. And so we'd really like to know what resource do you typically use to find somatic variant interpretations in cancer currently? So we've listed a lot of the commonly used databases, but if there's others that we haven't listed, you can select the other option. And again, it would be great if you could let us know in the chat what other databases or knowledge bases you use.
Okay, looks like the responses have stopped there. Oh, maybe a couple more coming in. All right.
All right, great. So ClinBar, Civic, and then OncoKB are the top three. All right, great. This is very useful. Thank you, everyone. So the somatic cancer Cdwg, we use Civic as our data curation platform because it is open access. It's a crowdsourced platform. And we've been collaborating with them right from the beginning. So there's several different features that they have introduced into the Civic system, specifically for the somatic cancer Cdwg activities. And one being we have different sub-organizations within the main ClinGen organization in Civic for all of our different curation activities. So pediatric cancer, hemologic, Janitor, Urinary, pH-like, as well as NTRAC. And you can find the curations and the members from each one of these organizations within these Civic within these Civic suborgs. To date, we have submitted 532 evidence items associated with 150 variants in our prioritized genes. And we've submitted 19 somatic variant assertions using the AMP-ASCO-CAP guidelines and 70 suggested sources for curation. Ooh. And we have about 93 of our members who have curator accounts in Civic. Now, these terms of evidence items and variant assertions, I'll go over some of them in the next few slides. But we also have a follow-up presentation on June 10th by Arpad Danos and Jason Saliba from the Civic team, and they will go in depth with what these different terms mean and the Civic data framework and structure, and how the somatic group curates variants in the Civic system. So Civic has incorporated the MVLD data format into their data structure as well. And this was done sometime in 2017, 2018. So all of MVLD's data elements are now mapped to Civic. And very briefly, in Civic, we curate an evidence item, so that's an EID, for a particular variant from a PubMed article. So from one PubMed article, we could curate one or multiple evidence items if the particular paper is talking about multiple variants, or the clinical significance, the role of that different clinical significance for that variant within the context of multiple diseases. So the variants can be, we can curate either the predictive, the diagnostic, or the prognostic effect of the variant in Civic in a given evidence item. So like I mentioned before, Civic is a crowdsourced system, and there's expert moderation in the platform. So when a curator submits an evidence item, it is submitted in the submitted stage. So that is that's shown in this orange color, and it has this orange tag on it, or flag on it. And then these the submitted evidence items are then moderated by a ClinGen Civic trained editor. And if everything is okay with the submitted evidence item, it goes into the accepted state which is shown in green. But if there's some issues with the evidence item and it is not aligned with our processes, then at that point of time, the editor would reject the evidence item, and that shows in red in the Civic system. Now, multiple evidence items. So again, evidence items are pieces of evidence from a publication that's related to a variant and the associated disease. And in Civic, we can summarize all of these evidences into a variant assertion. So the assertion is basically a summary of multiple evidences from different PubMed papers that are about clinical studies or preclinical studies and case studies and so on. And the assertion in Civic is then classified using the AMP-ASCO CAP levels of evidence. So briefly, I'll go over the somatic cancer variant curation process. So as I mentioned before, the somatic cancer Cdwg disease task forces will prioritize different genes and variants for curation. We then perform variant literature search, and this is in PubMed or Google Scholar. We look in ASCO abstracts, and there's also a couple of NLP tools that we're able to use, Civic Mine and Mace2K. And then a curator will curate and submit variant evidence in Civic from PubMed articles or ASCO abstracts. And these, again, are submitted as EIDs, so evidence items. So for example, if I come across a paper that shows EGFR L858R in NSC in non-small cell lung cancer supports sensitivity or response to the drug erlotinib, which is a tyrosine kinase inhibitor, then I would create an evidence item in Civic from that paper for this particular gene and variant. And then next, as I mentioned, we can also create assertions in Civic. So for EGFR L858R, we would summarize multiple evidence items that we've curated in Civic from different publications on this variant, and summarize all of that evidence into a variant assertion that is then classified using an AMP-ASCO CAP level of evidence. So in this particular case, EGFR L858R, this would get an AMP tier 1 level A evidence because erlotinib is FDA approved in non-small cell lung cancers. And there is a companion diagnostic associated with it as well. So next, once we submit the evidence items or the assertion into CIVIC, ClinGen and CIVIC editors would review the evidence submissions. So like I said, in the submitted state, the curation would show up in orange, and then in the accepted stage, it would show up, the particular entry would show up in green. And then finally, any outstanding issues with the variant interpretations are then discussed with the ClinGen Somatic Cancer Disease Task Forces and expert panels, and we arrive at a final conclusion. And the curations that are finally approved by the somatic expert panels will then be converted and then submitted to the ClinVar database. And I want to mention that we haven't submitted a fully approved variant assertion from our expert panels into ClinVar yet, but we've done some pilot submissions just to make sure that we're able to do that successfully. But hopefully, once NTRK Fusion receives the NTRK Fusion's SC VSEP receives a full approval from ClinGen, we'd be able to submit those curations into ClinVar in a 3-star expert panel status. So we have a small exercise for you all, and we'll also launch our poll questions for this, just to prepare you for the next session where we show you how we curate variants in the CIVIC system. But this particular exercise is just to sort of help you interpret a variant based on the AMP-ASCO-CAP guidelines. So here's one example. So a patient's diagnosed with late-stage melanoma, and tumor molecular profiling shows that this patient has the BRAF V600E somatic mutation. From a literature review, we see that melanoma patients with this variant, they predict sensitivity to a combination of dabrafenib and trametinib. And we know that that these two drugs are FDA approved for treatment of melanoma, melanoma patients with the BRAF V600E mutation. And this is also present in the NCCN guidelines. So at this point, we'll launch this poll and see how would you classify the BRAF V600E variant in melanoma, given this information. All right, I guess we can give it a few more minutes.
How is it? Yeah. Did you get to dance?
Okay. Looks like we have all the responses.
All right. Okay, so yes, the correct answer is Tier 1 Level A. It is AMP Tier 1 Level A, and this is because the two drugs are FDA approved for melanoma patients with BRAF V600E mutation, and this is also present in the NCCN guidelines. So that's why we would classify this variant as a Tier 1A variant with therapeutic evidence. All right, so moving on to the next Next case. So this is a pediatric cancer patient diagnosed with pilocytic astrocytoma. And the patient has, a BRAF fusion, so KIA 1549 and BRAF fusion. As per NCCN guidelines, we know that the presence of this fusion is reliable evidence that the tumor is a pilocytic astrocytoma. So what AMP tier or level of evidence would be assigned for this particular variant?
It looks like the responses have stopped.
All right, great. So the correct answer for this question is tier 1A diagnostic. And this is actually a variant that was curated by our pediatric disease task force. It's tier 1A because it is mentioned in the NCCN guidelines, which is, that's basically professional guidelines. It's mentioned in professional guidelines, and that's why we classify it as a tier 1 level A diagnostic variant. And then this is the final question. The final question here is, so again, this is in an in vitro study. It showed that the expression of a BRAF fusion, so the KIA 1549 BRAF fusion in pilocytic astrocytoma exhibits resistance to the drug bemorafenib due to activation of the MAP kinase pathway. And most of the evidence for this comes from preclinical studies. So how would we classify this variant?
It looks like the responses have stopped.
Okay, yep, and that is the correct answer. So level 2D therapeutic is the correct answer. And it's level 2D because most of the evidence supporting the clinical significance for this particular fusion comes from preclinical studies. And so that would be a level 2D evidence. All right, so with that, all of our somatic cancer variant curation training materials are available on our website. So on the website, you would click on the Curation Activities tab. Oops. The Curation Activities tab that would, and then in the drop-down, you would select somatic variant curation materials. And here you can click on training materials to view all of our tutorials, our video tutorials, as well as the MVLD publication and the standard operating procedures for curation in Civic. And you can also access Civic or sign up or sign into the Civic interface through our webpage. So I'll stop there. Like I mentioned, on June 10th, ARPAD Arpad Danos and Jason Saliba will go over the Civic platform and how we curate somatic variants in Civic. So there'll be more detail about the data framework used and the guidelines used and the interface in that follow-up presentation. So that we'd stop here. And if there's any questions for Deb and myself, we'd be happy to take them.
Thank you, Shruti, and thanks, Deb. That's just an amazing amount of work and just really interesting, I think, to think about the treatment and prognostic aspects of it as well. Does anyone have any questions? I did have a quick question just regarding how you prioritize the genes and variants that you are looking at.
So right now, they're actually just prioritized based on discussions with the task force members. So we essentially will first prioritize genes that are highly mutated or most commonly mutated in the cancer type of interest. And then the variants are essentially submitted by the different task force members based on, what they see in their internal databases or their patients. And then we create a list of variants that way and work on curating them.
Jenny, it's interesting because, the germline side, and we've talked about this in the somatic side, that germline kind of has this mandate by the secondary findings group, or at least, we've tried to, in the hereditary cancer group, cover a lot of the secondary finding genes related to cancer. We don't quite have that in the somatic space. So we have discussed, identifying some lists of, either common variants or problematic ones, but really we just haven't had that same centralizing list or gene list as we have with the germline.
Yeah, that's an interesting point, Deb. Yeah, I wonder even looking in ClinVar for VUSs and things like that, but I guess a lot of those are VUSs just because the information isn't there. But can you get additional information from members of your group maybe? And even information that's not published or?
Yeah, so it's very much informed by who's participating in the groups and the interest of those as well. And we try to, I would say, lead it some by looking at major cancer areas and trying to start to ensure that those are covered. I wouldn't, using ClinVar for it. There is somatic data in ClinVar. It is not primarily a somatic database, as we all know. It's more geared towards germline. So a lot of the somatic variants that would be in ClinVar, I'm not sure that using, unknown from ClinVar would lead us where we want. But yeah, any ideas on that is also good. We'd love to hear some feedback on that too, because it's something that's been an active question in the somatic working group and in the CVI.
And just to add to what Deb said, so in ClinVar, we actually just recently did a search, and there's only one submission of an NTRK fusion in ClinVar, and I think in total there's probably like 6 or 8 submissions of fusions in general. So like Deb mentioned, there's just very limited data, somatic data in ClinVar.
Well, especially for fusions. Fusions are pretty limited, just because of the submission, like the ability to submit it, or the data fields that they capture might not be as amenable to fusions. So yeah, that's a tough one.
Yeah. All right, any other questions or thoughts before we leave? It's just been really great to hear more about the work of your group. And the challenges that you come up against. Really great, and a lot of work, I can tell, and excited to hear from Arpad and Jason on June 10th for the Civic database discussion as well.
Great. Well, thanks for having us on.
Great. Thanks, Jenny. Thanks, everyone.
Thanks, everybody.
Bye.
Have a great day.
You too. Bye.